Welcome to EPG Pathshala. I am Kiran Kumar Salam, working as Assistant Professor in Hindu College, University of Delhi. Today, let us discuss the module Changing Concept of Gene. First, the learning objective of this module. After going through this module, you would be able to understand the concept of classical genetics, unit factor mentioned by Mendel, theory of genetics, that is, theory of dominant recessive genes, theory of independent assortment, and a theory of segregation given by Mendel. The concept of one gene, one polypeptide by Beadle and Tatum, the various types of genes, and lastly, the complementation experiment by Seymour Benja. The introduction. By 1885, the scientists were sure that the nucleus contained the hereditary material or information. The dilemma of germplasm and somatoplasm was made crystal clear by a classical experiment done by Augustus Weismann. He chopped the tail of mice for 22 generations and showed that it had no impact on the tail length of the progeny. Protein or enzyme coding region of the DNA are known as genes. Gene was coined in 1909 by Wilhelm Johansen. Gene theory was proposed by T.H. Morgan in 1911. In 1949, the role of genes in the protein synthesis was established by Linus Pauling and Vernon Ingram. The fine structure of gene was proposed in 1969 by Seymour Benja. Mendel's experiment acted as an initial study to provide information that the genes are the actual hereditary materials, which he called as the unit factor. These factors are passed from the parents to the progeny via the germ cells and are responsible for the transmission of the characters in the next generation. The discovery of genes or unit factor by Mendel was followed by various experiments providing evidences of them being the hereditary material. Sutton and Boveri gave the chromosomal theory of inheritance, that is, the genes are located on the chromosome. The studies of Morgan helped in understanding the concept of crossing over and creation of a genetic map. In addition to Sutton and Morgan, other scientists such as Bridges in 1923 and Mueller in 1927 and many others outlined the classical concept of gene, which include the gene positioning, crossing over, and their pattern of inheritance. The gene concept was modernized by Benja in 1957 by coining the terms histron, recon, and muton. He emphasized the different nature of genes and their location with respect to the inheritance pattern. Now let us understand the work of the Mendel. Gregor Johann Mendel is rightfully called as the founder of modern genetics. He was an Austrian monk. It took approximately 35 years for the people to recognize and appreciate his work, which formed the basis of modern genetics. Thus, he is recognized as the father of genetics. Around 1854, he worked with garden pea, Pisum sativum, and observed that the garden pea followed a particular pattern while passing on the trait from one generation to the another. He chose purebred, that is, plant whose traits remains the same in many generations. Pea plant was chosen because of its short lifespan, that is, it matured within one season and he could artificially hybridize them. He had successfully discovered and the basis of the transmission of hereditary trait by his meticulous experiments, mostly involving monohybrid crosses, and maintained accurate quantitative records. He gave the result using a simple statistical law and conveyed that these patterns were followed not only in the pea plant but also in the living beings having such traits. The trait he used for the experiment analysis was called the unit factor, which existed in pairs in individual organisms. Each unit factor came from either parent and the two forms were complementary to each other. For example, if we talk about the height of the plant, the traits were tall and short for the monohybrid cross, and the output consists of the three combinations, that is tall, short, and intermediate, as it is shown in the table 2. Until 1900, Mendel's experimental work was not appreciated by the scientist community and directly rejected when he presented his findings. However, three botanists, Hugo Debris, Erich von Schmarck, and Karl Correns, are credited for rediscovering the pioneering work of Mendel. Three of them worked independently and came to the same conclusion as Mendel. They interpreted their results in accordance to the Mendel's finding and drew attention to Mendelian studies on the pea. The classical genetics or unit factor by Mendel. Being a well-learned botanist, Mendel could identify characteristic traits which were opposite in two pea plants. Example, tall and short, smooth seed or wrinkled seed. Color of the seeds, whether yellow or green, shape of pod, wrinkle or smooth, pod color, yellow or green, flower position, whether terminal or axial, and the flower color, whether white or the purple, as shown in the figure 2. He cross-fertilized the pea plant with the opposite trait to obtain the progeny and recorded the observation. After analyzing his result, he concluded three important principles referred to as the laws of Mendel, or the Mendelian Principles, which were the law of dominance. This principle states that the one factor in a pair is a dominant over the other in inheritance unless the other factor in the pair are recessive. The progeny will express the dominant trait, shown in table 1. Law of segregation. The principle states that the offspring acquires one factor from its parent, and during the production of the gamete, the two copies of each hereditary factor segregates. The segregations of each factor is responsible for a combination of the trait from both the parents. Law of independent assortment. This principle states that the law of chance that governs which particular characteristics of the parental pairs will occur in each individual offspring. That is, the pairing and expression of the trait in the offspring are a chance factor. Figure 2 shows the diagrammatic representation of the study of the Mendel. After the experiment and its subsequent analysis, he concluded that the inheritance of this trait followed a basic classical pattern. He also added that this statistical pattern shown by the pea plant were also present in other organisms. His work was published in 1866, but as has been mentioned before, it remained unnoticed for a very long time until 1900. The significance of his work was appreciated and it came to be known as the Mendel's law. In the Mendelian times, the mechanism of the meiosis and the role of the chromosomes were not known. Mendel had concluded that there were units of inheritance which were responsible for transferring the parental trait to the offspring. With the advancement in the molecular biology, his postulates were welcomed and the field of genetics was discovered. Dominance or recessive. When a complementary unit factors, that is inherited from either parent, are present in a single progeny, one factor is a dominant unit factor which is able to express itself in the F1 progeny phenotype no matter what, and the other is known as the recessive unit factor which is not expressed in the phenotype of F1 progeny. We should remember that the term dominant or recessive are just designatory for individual trait in the parent's genotype. For example, if we consider the height of the plant, tall plants are considered the dominant over the recessive short or the dwarf plants. Segregation. Formation of the gametes lead to the segregation of the individual trait in order to equalize the chances for each trait in the individual gametes. Depending upon the fusion of the gametes, an individual may contain a pair of similar traits and the progeny will be decided accordingly. Because of segregation and a random fusion of the gametes, there is equal or 50% probability of receiving either of the trait. To understand it better, let us illustrate it through a monohybrid cross as shown in the table 2. Parent 1 is a tall and produces only capital T trait, whereas the parent 2 is a short and produces only small t trait. Consider the gamete of the parent 1, it will all be capital T unit factor, and for the parent 2 it will all be short or a small t unit factor. Following fertilization of the progeny will receive the one trait from its parent, that is the tall factor and a short factor. But because the tall factor is a dominant to the short, all the progeny are tall. On analyzing the gametes of the F1 progeny, its progeny has capital T, small t factors, which contributes to the tall height of the plant. However, on selfing the F1 progeny, we get four combinations in F2 progeny, which are tall tall, tall dwarf, dwarf tall, and lastly dwarf dwarf. A combination of 1, 2 and 3, which mentioned before, yield only tall plant, whereas the fourth one will yield short plant. Thus, we see that F2 progeny consists of both tall and short plants in a ratio 3:1. The ratio is well maintained in all the crosses for other traits observed by the Mendel. Mendel emphasized that it was these factors which were responsible for passing on the characteristics among the population from one progeny to the other. With time, these traits or unit factors were identified as individual alleles, and now we know that the person can be homozygous or heterozygous for a particular trait. We believe that a single trait was controlled by a single gene. However, modern genetics has revealed the concept of multiple alleles and the role of the environmental factors in expression of a gene. Mendel confined his work to the pea plant and explained the inheritance of the discrete hereditary factors, which are thereafter known as the genes. He laid the foundation of genetics, and later on it was found that his observations were true for a variety of the traits in many plants and animals. However, the explanations were not simple and easy for all inherited patterns, and a lot of complexities were observed by the later workers. Many of these workers even tried to resolve any of the complexities by following his principle with suitable modification. With time, they developed a lot of other patterns of inheritance and genetic expression, which are discussed below. Incomplete dominance. It is a form of the intermediate inheritance in which an allele specific for a particular trait is not able to express itself completely over its paired allele. As a result, the progeny expresses a phenotype having a combination of both the parental phenotype, that is an intermediate phenotype. This F1 phenotype is the result of the partial expression of both the parental allele. This is observed in the polygenic inheritance of traits such as the eye color and the skin color in human. In a snapdragon plant, Mirabilis jalapa or 4 o'clock plant, the parental phenotype of the flower is either red or white in color. But when red and white are crossed with each other, they get pink color in F1 progeny, as it is shown in figure 3. In this monohybrid cross, the allele which produce red color, capital R, is not completely expressed over the allele producing white color, small r. The resulting offsprings are pink in color. When these pink colored offsprings are self-pollinated, we get all the three phenotypes, that is red, pink and white, in a ratio of 1:2:1. However, when the F1 generation is a cross with either of the parental generations, we get equal number of the parent and F1 generation in the ratio 1:1, as shown in the figure 3. When pink flower in F1 generation is a cross with white flower, that is the parental generation, the F2 progeny contain both pink and white flower in equal proportion. The pink flowers are heterozygous with capital R small r genotype. The red and white flower are homozygous with capital R and small r genotype respectively. Codominance. In codominance, an additional phenotype is produced as both the alleles are expressed in the procedure. It is exhibited in the human blood groups ABO blood group type inheritance. Codominance in the human blood types, 4 different phenotypes are observed in the human with 3 different alleles, IA, IB and small I respectively. Both IA and IB are a dominant allele to the allele small I, that is, the IA and IB are codominant. This alphabet corresponds to a particular carbohydrate found on the surface of the person's red blood cells and give 4 different phenotypes, namely A, B, AB and O. Therefore, a person having a blood group A has the RBC coated with the carbohydrate A, and when both A and B carbohydrate are present, the blood group of the individual is the AB. Cells which are not coated with any of the carbohydrate are known to exhibit the blood group O. This is explained in a table 3. Because both A and B are a dominant allele, both express mutually and determine an individual phenotype. Differences between the incomplete dominance and codominance are shown in the table 4. Codominance is helpful in certain cases, example, sickle cell anemia. The disease is due to the presence of an abnormal protein, that is the beta chain of the hemoglobin has a substitution from the glutamic acid to valine at the 6th position. Normal hemoglobin is capital R, while the infected person has a sickle-shaped cells has a capital S. Carrier or heterozygous have a mixture of both normal and sickle-shaped cells and are produced when a healthy and infected persons interbreed, as shown in the figure 4. Sickle cell anemia is widely spread in the sub-Saharan African zone. These areas are prone to malaria, a disease caused by parasite Plasmodium falciparum and are transmitted through the mosquitoes. The heterozygous sickle cell trait has a moderately good resistance to malaria, as these deflated RBCs rarely developed the life-threatening anemia and the related problems observed in case of the homozygous S. The otherwise harmful sickle-linked allele provides an advantage for heterozygous individual. The homozygous R dominant have a normal RBCs which are excellent host for malaria. Hence, the nature selects in favor of the heterozygous sicklers against the homozygous sicklers with the normal RBC cells. The sickle cell mutation occurred thousands of years ago and helped to overcome malarial outbreak. The resulting survivors then pass on the mutation to their offsprings, and this trait becomes established throughout the sub-Saharan African region. Multiple allele. When a gene has 3 or more alternate forms of a same gene locus, it's known as a multiple allele genes. A well-known example is that of the human blood group again, where 3 alleles give rise to 4 phenotypes in the humans, as we discussed before in the example of codominance, table 4. Multiple alleles have a genetic benefit as they give a variety of the genetic combination and phenotypes. It is important to know our own blood group as it helps us to identify a suitable donor. If the blood is transfused from an incompatible donor, there are chances of a severe immune responses. As each blood group has its own specific type of the antibody, thus if we have a blood group A and receive a blood from a donor having a blood group B, our blood will produce antibodies and destroy the new RBCs having the carbohydrate B. Antibodies are specific protein produced within the body to identify and destroy the foreign invaders, whether it may be the protein, virus, etc. When a mismatched blood type appears, these antibodies bind to the unknown carbohydrate and kill the blood cells. Figure 5 explains this concept. Human genetics. The inheritance pattern of the diploid gene are same in human as observed in the bee and the Drosophila. But in the human breeding experiments are not very feasible. Usually, the information about the human inheritance is largely derived by studying the pedigree of naturally occurring traits. The identification of the trait might be of importance to human health and well-being, or help in understanding the health-related genetic defects. Knowing the nature of the defect or of those bearing it, that is the carrier, enables us to take appropriate steps to minimize its severity, and counseling can be given to reduce, if not eliminate, its propagations into the future generation. Various traits including blood groupings, eye color, hair texture, albinism, color blindness, ability to taste, PTC, that is phenylthiocarbamide, hemophilia, sickle cell anemia, inborn error of metabolism, and mental ability, and so on. Gene and metabolism. Let us discuss the Garretts classical experiment. Sir Archibald Garrett studied medicine at Oxford University. His father, Alfred Baring Garrett, was also a physician who studied rheumatoid arthritis. Garrett studied human disorder alkaptonuria by collecting family history and a urine sample. He deduced from this observation that alkaptonuria is a recessive genetic disorder, and in early 1900 introduced the phrase inborn error of metabolism. Here, tyrosine is not being broken down into carbon dioxide and water. He observed the accumulation of an intermediate product, that is the homogenesic acid, in the urine of the patient, which turned black when exposed to air, that is the oxidation of the homogenesic acid. He found that homogenesic acid was an intermediate product in the breakdown of the phenylalanine. The patient's leg enzyme homogentisate oxidase, which is responsible for converting the homogenesic acid into melyl isoacetate, as shown in the figure 7. He even published his studies in the book called The Incidence of Alkaptonuria, a study in chemical individuality in 1900, and states that the link between the genetics and the metabolic pathway. Garrett said that the genes are responsible for certain diseases like sickle cell anemia and Huntington's disease, alkaptonuria, cystinuria, pentasuria, and albinism are due to the defects in the metabolic pathway. That is, the genes determined the structure of the protein and enzyme. He proposed that the lack of the functional genes was responsible for these inherited traits or defects. He explained that certain defects are the result of the gene mutation in a biochemical pathway for eliminating the liquid waste. The change in the color of the urine is an indication of the error. In 1909, Archibald Garrett gave the concept of inborn errors of metabolism, which was proved by the studies of the George Beadle and Edward Tatum in 1941. Gene action refers to the expression of the genetic characters influenced by the gene. Enzymes are responsible for maintaining the homeostasis of the body cell through the process of autocatalysis. As a rule, one gene affects one enzyme. Color hormones, size, forms, production of the protein, antigens, antibodies decides its effect in the human diseases like albinism, tyrosinosis, etc. can be all linked to the defect in the gene. This idea was given by Beadle and Tatum in 1941, one gene, one polypeptide hypothesis. The idea of one gene, one enzyme was introduced in early 1940s and was proved by American geneticists George Wells Beadle and American biochemist Edward L. Tatum. They studied the growth of red bread mold Neurospora crassa. Their studies, often referred to as a Neurospora genetics, mainly involved growing mutant on a different media preparations, as you can see in the figure 9. The mold Neurospora was the first exposed to X-ray to obtain various mutations. The mutations are the sudden heritable change in the genome of the organism having the capability of disrupting the mold's ability to synthesize the amino acids or the nutrients and grow. They subsequently grow the mutant spores in the tubes having complete medium, that is amino acid, vitamins and organic substances along with the proper physical environment. Then these mutant strands are grown on minimal growth media containing only the basic nutrients in which the wild type or the non-mutated strand of the neurospora could survive. They observed that the mutants were not able to grow on the minimal medium and required an additional supplement of specific amino acid in order to grow. The effect of the mutation was identified by adding the supplements one by one to test which supplements could fulfill the requirement of the missing enzyme to grow. Surprisingly, these mutants, when crossed with the normal molds, were able to grow in the minimal medium, revealing that these metabolic defects were outcome of the recessive trait and proved the alterations in the genes, as it is shown in the figure 10. Thus, it was concluded that the mutants have disruption in the specific genes which was responsible for production of a specific amino acid. An identifiable nutritional mutation with X-ray technique was achieved in their 299th attempt when it can grow in a minimal medium supplemented with vitamin B6. Thus, with patience and continued perseverance, they were able to identify many more mutants, revealing that genetic mutations affect metabolic pathway and give the detailed multi-step synthesis pathway for many amino acids and vitamins. The information obtained from the experiments on the neurospora was confirmed when Mendel witnesses it in a Drosophila when he worked with the Ifrosi. They published their result in Genetic Control of Biochemical Reactions in Neurospora in 1941, proposed the concept of one gene, one enzyme hypothesis. This hypothesis was verified, but since 1940s it has undergone a slight change, and today it is known as the one gene, one polypeptide hypothesis as stated by Yanofsky. One gene, one polypeptide hypothesis states that a single gene is responsible for coding a single polypeptide. A gene consists of a specific sequence of the nucleotide, a triplet code for an amino acid which forms the monomeric unit of the polypeptide joined to each other by a peptide bond. Earlier, it was known that a single gene is responsible for the polypeptide. In a laboratory, the fungus Neurospora crassa grows on a minimal medium containing agar, sucrose nitrate, vitamin, that is biotin, and inorganic minerals. This means that all the supplements are present within this minimal media for the fungal development. However, when treated with the X-ray, the fungus is unable to grow in the minimal media. The mutant spores on the minimal media are supplemented to determine the deficient component which the mutants are unable to synthesize. The mutants give a healthy progeny when crossed with the normal or wild type, and the ascospores of the progeny are individually tested for their nutritional requirement. A single gene mutation is determined crossing the single arginine deficient mutant strand A negative with the wild type A positive. All the 8 ascospores of the progeny survived on the medium containing the arginine, but only 4 ascospores survives in absence of the arginine from the medium. The mutation of the gene leads to the suppression of the specific steps. This is demonstrated by growing of mutants on the minimal medium when the deficient component is added to it. Thus, on identifying the mutants, we know the defect in the biosynthetic pathway. This conveys the basic enzyme relationships. Currently, the concept of one gene, one enzyme has been revised to one gene, one polypeptide chain. In view of the complexity in the structure, functions of the enzyme and the genetic variability of the protein, such as in case of the sickle cell hemoglobin. However, later on, realizing the phenomenon of splicing with which it became evident that a single gene codes for a specific non-enzymatic protein as well as the individual polypeptide chain. Now, we know that certain genes code for the different types of RNA involved in protein synthesis. Especially in the eukaryotes, a single gene can produce several RNA products depending on the differential splicing. Hence, it was modified to one gene, one polypeptide hypothesis. Now let us see the gene type on the basis of their behavior. A variety of the gene types have been proposed while studying the behavior of genes and interaction among them, such as lethal gene, pleiotropic gene, inhibitory genes, etc. Details is illustrated in table 5. And again in table 6 gives the genes in the protein synthesis, such as the structural genes, operator genes, regulator genes. The neoclassical concept of genes. Identification of a gene and a genetic material done by the different geneticists, which brought a blow to the classical concept of the genes, are given in table 7. Frederick Griffith had discovered transformation in 1928. He had revealed that kill the bacterial cells when injected into the healthy mice were able to transform genetically into the living bacteria. This is illustrated in figure 11. Avery et al. In 1944 proved that substances responsible for transformation in the bacteria was the DNA, as the transformation failed to occur when the sample was treated with the DNase or deoxyribonuclease, as shown in the figure 12. Thus, it was concluded that the DNA consists of genes. Hershey and Chase in 1952 were able to prove that the only component of the bacterial viruses or bacteriophage was its DNA, which enters the bacterial host by radioactively labeling the protein sulfur 35 and the DNA phosphorus 32. And therefore, it was presumed that DNA is the sole bearer of the viral genetic information. The experimental evidence that DNA is the viral genetic material is demonstrated in figure 13. Here in the table 7, the names of some of the scientists and their observations and classical views of the genes are explained. Modern concept of gene proposed by Seymour Bender. 20th century has witnessed a lot of development in the biological sciences. It began with the Mendelian units of hereditary to the entire human genome. However, Seymour Bender's studies played a very important role in shifting the genetic studies from the classical to the advanced molecular genetics. Even though he graduated in physics from Purdue University, he had a lot of interest in biology after reading the book What is Life? Bender took the summer course on bacteriophages at Cold Spring Harbor Laboratory in 1948. In 1953, after Watson and Crick published their model of DNA, Bender hatched his plan to get inside the gene by using the bacteriophage. He won the American Nobel Prize, that is the Albany Medical Center Prize in Medicine and Biomedical Research in 2006 for the behavior change in the Drosophila due to a single gene mutation. Let us deep into his experiment on a cystone concept. Complementation assay, the Bender studied the bacteriophage T4 genome having a mutations on the R2 gene. The mutants have a distinct host range and a plaque morphology. Growing E. coli cells, when infected with the wild-type T4 phage, produce a small and a turbid plaque with the fuzzy ends, in contrast to the plaque proposed by R2 mutant, which are the smaller in size and possess a distinct boundary. Wild-type T4 phage has an ability to form plaque on E. coli strand B and the E. coli strand K12 lambda, whereas the R2 mutants were able to grow only in the E. coli B strand. E. coli K12 strand lambda was not infected by the T4 phage. This property of the different plaque structure was used by Bender to isolate a small proportion of the wild-type phage among the large proportion of the R2 mutant. Figure 15 shows the complementation experiment showing two possible outcomes. No complementations occurs since the co-infected cells is unable to make the normal product of the gene A. The co-infected cells will not produce the viral particles, thus no bacterial cell lysis and hence no plaque formation occurs. Complementation occurs since the co-infected cells is able to make the normal product of gene A and gene B. The co-infected cells will produce the viral particle that lyse the cell, resulting in the appearance of a clear plaque. Take a moment and observe the experiment shown in the figure 15. He was able to identify 60 different R2 mutants which he crossed in all the possible combinations using E. coli as the host. He collected the progeny after the cell lysis, considering X and Y as the two R2 mutants. Bendel crossed R2X with the R2Y, and the progeny obtained consists of the two parental type R2X and R2Y. The two combination type R2X, R2Y, that is the double mutant, and the wild type is only R2. His observation revealed approximately equal number of the parental and recombinant in the progeny. However, the relative the frequency of the parental and the progeny population was dependent on the distance between the two alleles. He plated the phage progeny on the permissive host, that is E. coli B, and then from the total number of the plaque produced, he calculated the total number of the plaque progeny per milliliter. Later on, he plated another sample of the plaque T4 on the non-permissive host E. coli K12 lambda and found the frequency of the R plus recombinant. Thus, he was able to find the percentage of the very rare R plus recombinant produced by crossing over between closely linked alleles. The recombination frequency was given by the formula 2 times the number of the R plus recombinant divided by total number of the progeny into 100%. The number of R plus recombinant is multiplied by 2 to account for the double mutants. A control was set up for each cross in which R2 parent was used to infect the E. coli B, and later the progenies were used to infect both E. coli B and K12 lambda separately. This was done to calculate the reversion frequency because the changes are that R2 mutant undergoes a mutation to revert back to the R plus type. Bendel concluded that the reversion frequencies were a much lower magnitude than that of the smallest recombination frequency. He constructed a linear genetic map from the recombination data obtained from the 60 R2 mutants. R positive recombinant were not obtained from the certain crosses, which revealed that these pairs carried the mutates at exactly the site. Thus, homoallelic mutations were responsible to change the same nucleotide pair within a gene. Heteroallelic mutations were able to change different nucleotide pair within a gene. Till now, it was believed that a gene is a functional unit of DNA and each gene has a specific role. Bendel devised an experiment to find whether the two different R2 mutants had the same function. Bendel adopted the cis-trans test or complementation test given by Edward Lewis to study the nature of the gene in Drosophila as shown in figure 16. Complementation test showed that R2 region consists of the two genes, namely A and B. Any mutations within these two regions result in the R2 mutants, having a specific plaque morphology and host range property. By this, we can say that R2A and R2B give different products when grown in the E. coli K12 lambda. Complementation test helps us to identify the units of the genes which are having a specific mutation leading to the expression of the same mutant phenotype. Bendel deliberately infect the non-permissive host E. coli K12 lambda with a pair of the R2 mutant phage to see whether they are able to survive together and produce the progeny phage. If a progeny are obtained when the paired mutants are used to infect the non-permissive host, it conveys that the mutants are complementary to each other. When both R2 mutants are present, the combined product allows the progeny to propagate. In the absence of the progeny, the mutants are not complementary, indicating the presence of the mutations on the same functional gene or units. That is, the life cycle of the phage cannot proceed because both the mutants are producing the defective genes and no progeny is observed. S. Benjor in 1957 coined the specific terms of the genes depending on their nature of the genetic materials in relation to their locations on the chromosome and the basis of the genetic phenomenon to which they involve. Benjor reported the fine structure of a gene basis on its property like the ability to mutate, recombine, and the specific functions on the gene. He classified the gene into three structures: cistron, muton, and a recon. A cistron as a unit of the transmission. It is a part of the DNA specifying a single polypeptide chain. A cistron can have 100 nucleotide pair in length to 30,000 nucleotide pairs. It transmits the characters from one generation to the other as a result of the transmission. Recon as a unit of the recombination. It is the smallest segment of the DNA capable of being separated and exchanged with other chromosomes. A recon consists of not more than 2 pairs of the nucleotide. Muton as a unit of mutation. Muton is the smallest unit of the genetic material which, when changed or mutated, produce a phenotypic trait. Thus, muton is delimited to a single nucleotide. Having gone through these modules, let us now discuss the summary. Gene as the coding region of the DNA which are transcribed to give a different functional protein or enzyme according to the needs of the cell. Mendel used the garden pea as his experimental model and gave the 3 Mendelian principles, which were the law of dominance. The progeny expresses the dominant trait, law of segregation. The segregation of each factor is responsible for the combinations of the trait from both the parents. Law of independent assortment. The pairing and expression of the trait in the offsprings are a chance factor. Incomplete dominance. The progeny expresses a phenotype having a combination of both the parental phenotype, that is, an intermediate phenotype. Codominance is observed when both the alleles are dominant and expresses in the progeny. Example, ABO blood group of the humans. If more than 2 alleles are responsible for a particular gene, the concept is known as the multiple gene. Sickle cell anemia has not eliminated by the natural selection because of the advantages to the heterozygous individual. Certain diseases like Huntington's disease, alkaptonuria, cystinuria, pantosuria, and albinism are due to defects in the metabolic pathway. Neurospora crassa involved growing the mutants on a minimal medium and realized that the mutants needed additional supplements of a specific amino acid in order to grow. One gene, one polypeptide hypothesis states that a single gene is responsible for coding and the productions of the single polypeptide. Seymour Benjer's complementation experiment developed in 1950s studied the substructure of the gene. Lastly, genes are now classified into 3 structures: cistron, muton, and recon, based on its properties like the ability to mutate, recombine, and the specific functions of the gene. Thank you and best of luck.